Hi, welcome back. I'm Dr. Chengzi the Orange. In our last video, we explored how radiation uses powerful beams of energy to destroy cancer. Today, we're moving to a completely different battlefield, drug-based cancer therapy. This is one of the most exciting areas of cancer treatment and one of the fastest evolving. It includes chemotherapy, hormone therapy, targeted therapy, and immunotherapy. To patients, these treatments often look surprisingly ordinary. A plastic IV bag dripping clear liquid, a syringe or a bottle of pills, with names almost impossible to pronounce. But beneath that ordinary appearance, a microscopic war is taking place, influencing how billions of cells grow, communicate, divide, mutate, and survive. These treatments didn't appear overnight. For most of human history, from the ancient Egyptians and Greeks to the late 1700s, people believed cancer was caused by an imbalance of the body's fluids, known as the four humors. If disease came from abnormal body fluids, treatment focused on restoring balance rather than attacking the disease itself. Doctors relied on special diets, bloodletting, laxatives, and herbal remedies. Aside from occasional surgery or cauterization, there was little they could actually do. The turning point came in 1838 when German scientist Johannes Müller examined tumors under an early microscopes and reached a revolutionary conclusion. Cancers arise from cells but not from body fluids. That discovery changed everything. For the first time, scientists knew where cancer came from. They could begin to study and eventually target cancer cells, chromosomes, microtubules, DNA, RNA, enzymes, signaling pathways, and even the complex ecosystem surrounding a tumor. That single discovery laid the foundation for every modern cancer drug we use today. Now let's begin with chemotherapy, the large-scale bombing campaign against cancer. Modern chemotherapy began in the 1940s. Before then, some of the first cancer medicines were actually poisons. Arsenic compounds like Fowler's solution were used to treat certain cancers despite severe side effects. But when there were no better options, doctors accepted the risks in the hope of saving lives. Today, chemotherapy includes many different drugs, each designed to attack cancer in a different way. Based on how they work and where they come from, these drugs can be grouped into 5 major categories: alkylating agents, antimetabolites, natural product-derived antimitotic agents, anti-tumor antibiotics, and other cytotoxic agents. One of the most fascinating origin stories in cancer treatment begins not in a hospital, but on the battlefield. During World War I, armies used a deadly chemical weapon called mustard gas. Soldiers exposed to it suffered severe burns, but doctors also noticed something unexpected. Their bone marrow and white blood cells were almost completely wiped out. At the time, this was simply viewed as another horrifying consequence of chemical warfare. More than 2 decades later, during World War II, scientists began asking a very different question. If mustard gas could destroy rapidly growing white blood cells, could a safer, modified version be used to destroy rapidly growing cancer cells as well? In 1942, pharmacologists Louis S. Goodman and Alfred Gilman, working with surgeon Gustav Linskog, secretly tested a modified compound called nitrogen mustard on a patient with advanced lymphoma. The patient had a massive tumor that made breathing difficult. Within days, the tumors shrank dramatically, and the patient could breathe more easily. Although the cancer eventually returned, this was the first convincing demonstration that a drug could make cancer regress. That remarkable success marked the birth of modern chemotherapy. Scientists soon realized that nitrogen mustard worked by attaching chemical alkyl groups to DNA, preventing cancer cells from copying their genetic material and dividing. This led to the development of an entire family of drugs known as alkylating agents. It is one of medicine's greatest ironies. A chemical weapon created for war ultimately became the foundation of one of the world's most powerful weapons against cancer. From 1940 to 2024, approximately 20 alkylating agents have been approved by the FDA or the European Medicines Agency. Well-known examples include cyclophosphamide, melphalan, temozolomide, busulfan, and lomustine, which are widely used to treat lymphomas, leukemias, ovarian cancer, multiple myeloma, and brain tumors. Notably, several early alkylating agents, such as cyclophosphamide, melphalan, busulfan, and chlorambucil, remain mainstays of cancer therapy today. In the 1940s, biology was entering the new era of molecular biology. Scientists had discovered that every cell must faithfully copy its DNA before it can divide, and that DNA is built from chemical building blocks called purines and pyrimidines. They also realized that rapidly growing cancer cells consume these building blocks at an extraordinary rate. This sparked a revolutionary idea. Instead of trying to poison cancer cells directly, what if scientists could trick them into using counterfeit building blocks? Once incorporated into DNA or RNA, these imposters would sabotage nucleic acid synthesis and bring cell division to a halt. In 1947, researchers at Liddell Laboratories had just succeeded in producing large quantities of folic acid, a vitamin essential for making DNA and new blood cells. Hoping it might help children with acute lymphoblastic leukemia, Sidney Farber, a pathologist at Boston Children's Hospital, administered folic acid to his young patients. Instead of helping, the leukemia progressed even faster. Farber realized that the cancer cells were feeding on folic acid to fuel their explosive growth. He reasoned that if folic acid accelerated leukemia, then blocking its action might have the opposite effect. Fortunately, scientists at Liddell Laboratories, including biochemist Yellapragada Subbarao, had already developed aminopterin, a compound that closely resembles folic acid but prevents cells from using it. Farber treated 16 children with acute leukemia using aminopterin. At a time when childhood leukemia was almost always fatal, the results were astonishing. 10 of the 16 children achieved temporary remission. Although the remissions were short-lived, they launched the era of anti-metabolite chemotherapy and rational drug design. The success of aminopterin inspired scientists to create many more anti-metabolites that mimic the cell's natural metabolites but disrupt their normal function. Instead of supplying cancer cells with the raw materials they need, these drugs hand them counterfeit building blocks, causing DNA or RNA synthesis to fail. As of 2024, 18 anti-metabolite drugs have been approved by the FDA or EMA. Among them, 6 mercaptoprine and 5 fluorouracil, both introduced in the early 1950s, remain cornerstone treatments for a wide range of blood cancers and solid tumors. The discovery of antimitotic drugs was another remarkable example of scientific serendipity. In the 1950s, Canadian scientists Robert Noble and Charles Beer were investigating the Madagascar periwinkle, a colorful flower long used in traditional folk medicine to treat diabetes. They weren't looking for a cancer treatment, but during animal experiments, they stumbled upon an unexpected finding. The plant extracts caused profound bone marrow suppression and a dramatic drop in white blood cells. To most researchers, this would have seemed like an alarming side effect. But Noble and Beer saw an opportunity. Since leukemia is a cancer of white blood cells, they wondered, could this mysterious plant be selectively suppressing leukemia cells? Working with Eli Lilly and Company, they isolated 2 remarkable compounds, vinblastine and vincristine. Both proved highly effective against leukemia and lymphoma, becoming some of the earliest successful plant-derived chemotherapy drugs. Scientists later discovered how they worked. Inside every dividing cell are microscopic protein fibers called microtubules, which function like ropes pulling duplicated chromosomes apart before the cell splits in two. Vinblastine and vincristine prevent these ropes from ever forming. Without them, cell division comes to a standstill, and the cancer cell dies. The success of these drugs sparked an ambitious new mission. In 1960, the National Cancer Institute partnered with the United States Department of Agriculture to search nature for more anti-cancer compounds. Over the next 2 decades, researchers collected and tested more than 114,000 plant extracts from around the world. It became one of the largest natural product drug discovery programs ever undertaken. That enormous effort uncovered several future cancer-fighting superstars. From the bark of the Pacific yew tree came paclitaxel, better known as Taxol. From the Chinese happy tree came camptothecin, and from the mayapple plant came podophyllitoxin. These discoveries later gave rise to multiple FDA-approved cancer drugs that continue to save lives today. Interestingly, not all of these drugs work the same way. Vinblastine and vincristine prevent microtubules from forming, while paclitaxel does exactly the opposite. It locks microtubules in place, preventing them from disassembling. Either way, the chromosomes become trapped, and cell division grinds to a halt. Camptothecin and podophyllitoxin attack a different target altogether. They inhibit topoisomerases, enzymes that normally untangle DNA during replication, causing DNA replication to fail and rapidly dividing cancer cells to die. Although many of these early natural compounds were highly toxic, they opened an entirely new frontier in cancer therapy. Today, at least a dozen FDA-approved microtubule-targeting drugs, along with numerous semi-synthetic derivatives, trace their origins back to these discoveries. By the mid-20th century, antibiotics had already revolutionized medicine. Drugs like penicillin were saving millions of lives by killing bacteria. Their success inspired scientists to ask a simple question. If microorganisms can make chemicals that kill bacteria, could they also make chemicals that kill cancer cells? Researchers began testing thousands of compounds produced by soil bacteria and fungi. Many turned out to be too toxic to use as antibiotics, but that failure became an unexpected success. Because cancer cells divide so rapidly, some of these highly toxic compounds were especially effective at destroying them. This led to the discovery of a new class of chemotherapy drugs called anti-tumor antibiotics. Most of the drugs in this category attack the DNA or RNA of the cancer cells, preventing them from growing and dividing. Today, drugs such as doxorubicin, daunorubicin, and bleomycin remain important chemotherapy drugs for cancer treatment. Not every chemotherapy drug fits neatly into the 4 major categories. Some are simply grouped as other cytotoxic agents. Their discoveries remind us that breakthroughs can come from the most unexpected places and sometimes by pure chance. Take cisplatin, for example. In the 1960s, Barnett Rosenberg at Michigan State University noticed that platinum released from laboratory electrodes had stopped bacteria from dividing. That unexpected observation eventually led to the development of cisplatin, which became one of the most successful chemotherapy drugs in history. Fortune struck more than once. Asparaginase emerged after scientists found that guinea pig serum unexpectedly shrank lymphomas in mice, while hydroxyurea sat unnoticed for nearly a century before its anti-cancer potential was finally recognized. As Louis Pasteur famously said, "Chance favors the prepared mind." Now we start talking about hormone therapy on cancers. For decades, doctors believed that the only way to fight cancer was to poison it. But in 1896, Scottish surgeon George Beatson observed something remarkable. After removing the ovaries of women with advanced breast cancer, some of their tumors shrank. It seemed that breast cancer cells depended on mysterious chemicals produced by the ovaries. Without them, the cancer cells were starved. A few decades later, doctors discovered that prostate cancer could also regress after surgically removing the testicles. The pattern was strikingly similar in these male and female specific cancers. At the time, however, no one knew what these mysterious chemicals actually were. Ironically, extracts from ovaries were already being used to relieve menopausal symptoms, while extracts from animal testes and even preparations made from male urine were being marketed as rejuvenation therapies. In the 1920s and 1930s, scientists finally isolated estrogen and synthesized testosterone, revealing the powerful chemical messengers behind these remarkable effects. In 1941, Charles Huggins discovered that prostate cancer depends on male hormones for growth and showed that lowering testosterone through surgical castration or high-dose estrogen could shrink advanced tumors. This breakthrough founded modern hormone therapy and earned him the 1966 Nobel Prize in Physiology or Medicine. Today, hormone therapy is far more precise. Instead of removing organs or giving large doses of opposite-sex hormones, doctors use drugs that selectively block hormone production or prevent hormones from activating cancer cells. Medicines such as tamoxifen, anastrozole, letrozole, leuprolide, degarelix, and enzalutamide have transformed the field. More than a century after its surprising beginning, hormone therapy remains one of the most effective and widely used treatments for hormone-dependent cancers, particularly breast and prostate cancer. Now, time to start talking about targeted therapy. Targeted therapy is one of the biggest success stories in modern cancer medicine. According to the American Cancer Society, it is a form of precision medicine that attacks specific molecules or mutations found in cancer cells. Sounds impressive. But what does that actually mean? Imagine your body as a kingdom and tumors are terrorists-occupied regions. Chemotherapy is like carpet bombing, effective, but it inevitably damages many healthy buildings along with the enemy. Targeted therapy is different. It's like sending a GPS-guided missile. Instead of attacking every rapidly dividing cell, doctors first identify the cancer's unique weaknesses, its genetic mutations, abnormal receptors, overactive signaling pathways, or even the blood vessels feeding the tumor. Then they choose a drug designed to hit that specific target while leaving most normal cells alone. Of course, doctors couldn't do this until they knew what the targets were and how to build the missiles. That's why targeted therapy is a relatively recent breakthrough. It took decades of scientific advances to make it possible. The first breakthrough came from the genetics revolution. As DNA sequencing became possible in the 1970s and 1980s, scientists began uncovering the genetic blueprint of cancer. They discovered oncogenes such as RAS and HER2, which can drive cancer when switched on, and tumor suppressor genes such as p53, which normally protect us from cancer but allow tumors to develop when they are lost or damaged. The completion of the Human Genome Project in 2003 marked another giant leap forward. It was soon followed by large-scale cancer genome projects that decoded the genomes of thousands of tumors, revealing the genetic mutations responsible for many cancers. For the first time, scientists had a comprehensive catalog of cancer-causing mutations, the very targets that precision missiles could be designed to attack. The second breakthrough was understanding how those mutations change the behavior of cells. Scientists began uncovering cellular signaling pathways in the late 1950s, but it wasn't until 1982 that the first major cancer signaling mechanisms were understood. Researchers discovered that mutations in genes such as RAS can permanently switch these pathways on, continuously driving cells to grow, divide, and survive. Since then, dozens of cancer signaling pathways have been identified, and the faulty proteins within them becoming ideal targets for precision medicines. The third breakthrough was the invention of monoclonal antibodies in 1975, which act like highly precise guided missiles. They recognize a single target on the surface of cancer cells and lock onto it with remarkable accuracy. Once attached, these antibodies can destroy cancer in several ways. They can block the growth signals that cancer cells depend on, flag the cancer cells for destruction by the immune system, deliver toxic chemotherapy drugs directly to the tumor, or even cut off the tumor's blood supply and starve it. Better yet, monoclonal antibodies can be mass-produced in laboratories rather than harvested from living animals, making them practical for widespread clinical use. In 1997, the first monoclonal antibody cancer drug, rituximab, was approved, marking the beginning of a new era in targeted cancer therapy. The fourth breakthrough came from advances in rational drug design and the development of small molecule drugs. Although the concept emerged in the 1960s, it flourished in the 1980s. Instead of randomly screening thousands of chemicals in cells and animals, scientists could now use structural biology and computer modeling to design molecules that fit abnormal proteins with remarkable precision. This rational approach dramatically increased the efficiency of drug discovery, reducing years of trial and error and greatly decreasing the need to test countless compounds in laboratory cells and animals. These small molecules became another class of precision-guided missiles, filling a critical gap left by monoclonal antibodies. Because antibodies are too large to enter cells, they can only attack targets on the cell surface. Small molecules, however, are tiny enough to cross cell membranes and disable abnormal proteins hidden inside cancer cells. This breakthrough produced one of the most celebrated targeted drugs in history, imatinib. Approved in 2001, it transformed chronic myeloid leukemia from a once fatal disease into one that many patients can successfully keep under control for decades. Today, targeted therapy has become one of the pillars of modern oncology. More than 90 FDA-approved small molecule targeted drugs, together with dozens of monoclonal antibodies, are used to treat cancers driven by specific genetic alterations. By now, we've seen several ways to attack cancer directly. Surgery removes it. Radiation destroys it with high-energy beams. Chemotherapy poisons rapidly dividing cells. Targeted therapy strikes specific molecular weaknesses. But all of these weapons come from outside the body. Can we fight cancer from within? Yes, we can, by harnessing our own immune system. Cancer immunotherapy actually began decades before scientists even understood the immune system. In the early 1890s, American surgeon William Coley noticed that a patient with advanced sarcoma experienced a remarkable tumor regression and eventually became cancer-free after developing erysipelas, a bacterial skin infection. Coley wondered whether the infection had somehow stimulated the body's defenses to attack the cancer. To test this idea, he created Coley's toxin, a mixture of heat-killed bacteria that was injected into patients with sarcoma. In 1899, Coley's toxin was commercialized and remained in clinical use for nearly 70 years. However, the treatment often triggered powerful immune reactions, including high fever, severe infections, and occasionally death. As chemotherapy emerged in the mid-20th century, concerns about its inconsistent effectiveness and safety led to its gradual abandonment. Decades later, scientists finally realized that Coley had unknowingly pioneered the world's first cancer immunotherapy. By the 1980s, immunology had entered a new age. Scientists had mapped hundreds of immune cells, antibodies, receptors, and signaling pathways, revealing an elaborate police and military network that protects our bodies. With this knowledge, they hoped to enlist the immune system in the fight against cancer. Researchers first tried to strengthen the immune army with powerful signaling molecules such as interferon and interleukin-2. The strategy worked spectacularly in a small number of patients. Some achieved complete remissions lasting for decades. But most patients did not respond, while many experienced severe toxicity because the treatment activated immune cells throughout the body. The lesson was not that immune stimulation failed. It was that the immune system needed a more precise and controllable strategy. The real breakthrough in cancer immunotherapy came when scientists discovered immune checkpoint proteins and developed drugs that allow the immune system to fight cancer more effectively. Using melanoma as an example, imagine the body as a kingdom. Cancer cells are like terrorists, while a nearby lymph node is a military camp. Dendritic cells serve as intelligence officers, and naive T cells are rookie soldiers. As the tumor grows, nearby normal cells become stressed and send out distress signals. Dendritic cells are dispatched to collect tumor antigens and present them to naive T cells in the lymph node. A naive T cell is activated only when both its T cell receptor, TCR, and CD28 receptor receive the proper signals. Once activated, it rapidly proliferates, forming a large army of killer T cells ready to attack the tumor. To prevent an excessive immune response, activated T cells produce CTLA-4, a checkpoint protein that acts as a brake on further activation and proliferation. Cancer cells can exploit this natural brake by increasing CTLA-4 signaling, weakening the immune response. Scientists developed CTLA-4 inhibitors to block this brake, allowing T cells to continue multiplying to fight the tumor. The story is not over yet. With the help of CTLA-4 inhibitors, the T cell army completes its training and assembles in the lymph nodes. The army then travels through the bloodstream and surrounds the tumor. Now the killer T cells use their T cell receptors to precisely recognize cancer cells and launch their weapons, powerful enzymes that penetrate and destroy the target cells. Will the cancer cells simply accept defeat? Of course not. They are highly cunning. They can display PD-L1 on their surface, which binds to the PD-1 receptors on T cells. This interaction acts like a fake peace do not fight order, suppressing the T cell's attack. To overcome this trick, scientists developed PD-1 and PD-L1 inhibitors. These drugs block the false no-fight signal, allowing T cells to ignore the deception and continue the mission of destroying the tumor. This table shows today's major checkpoint inhibitors. Don't memorize them, that's scary. Just remember, if cancer ever comes, modern medicine has many weapons ready to fight. But cancer is still an extraordinarily clever enemy. Some cancer cells don't just press the brakes. They hide their identity altogether by removing the molecular ID badges that T cells normally recognize. To overcome this trick, scientists developed one of the most remarkable advances in modern medicine, CAR T-cell therapy. Doctors first collect a patient's own T cells and genetically engineer them to express a synthetic receptor, essentially installing an AI-powered target acquisition and missile guidance system that continuously scans for a specific cancer marker, locks onto the enemy, and directs a precision strike. The upgraded T cells are then expanded into hundreds of millions of elite immune soldiers before being infused back into the patient. These CAR T cells hunt down and destroy cancer, even when it tries to hide from the body's natural immune defenses. Today, CAR T-cell therapy has produced remarkable results against leukemia, lymphoma, and multiple myeloma, while engineered NK cells, macrophages, and many other forms of immunotherapy are rapidly expanding the immune system's arsenal against cancer. Like every powerful weapon, however, immunotherapy comes at a price. By releasing the immune system's safety brakes, checkpoint inhibitors can sometimes make the immune police too aggressive, causing them to attack healthy organs. This can lead to autoimmune-like side effects affecting the skin, intestines, lungs, liver, thyroid, and other tissues. CAR T-cell therapy can also trigger serious complications, including cytokine release syndrome, a massive immune overreaction resembling a kingdom-wide emergency alarm, as well as neurological side effects. Nevertheless, immunotherapy has transformed cancer treatment and remains one of the most exciting and rapidly advancing frontiers in modern medicine. So, with so many cancer drugs available, how do doctors choose the right ones? It comes down to 4 basic rules. First, know the cancer, what type it is, where it is, and how far it has spread. Second, find its weak spots. Biomarker and genetic testing can reveal specific targets that certain drugs can attack. Third, know the patient. Age, overall health, previous treatments, and possible side effects all matter. And fourth, choose the best strategy. Chemotherapy, targeted therapy, immunotherapy, hormone therapy, or often a combination of them. Cancer treatment is no longer one size fits all. It's about finding the right treatment for the right patient at the right time. That's personalized cancer medicine. That brings us to the end of today's talk. In our next episode, we'll tackle two questions nearly every cancer patient fears. Why does cancer come back? And is cancer always worth treating? Thank you for watching. See you soon.